All right, let's talk about a pathogen that you've probably seen but might not know the whole story behind. Trueperella pyogenes. If you're a medical microbiologist who's ever looked at one of these plates and thought, okay, what's really going on here? Then this deep dive is for you. We're going to cover it all, from the bench to the bedside. You see that classic, wide, beautiful zone of beta-hemolysis on the blood agar, and your brain immediately starts running through the possibilities. But with Trueperella pyogenes, that's just the tip of the iceberg. Behind that hemolysis is a crazy history of identity crises, and more importantly, a huge gap in antimicrobial reporting that directly impacts patient care today. So, let's start with its identity crisis. I mean, you really have to understand this bug's complicated past to get a grip on it. And this isn't just a boring history lesson, I promise. Knowing its old names is actually critical for making sense of older studies, or even digging through your own lab's historical data. I mean, just look at this timeline. This organism has been bounced around more than a pinball. It started as a bacillus, then got moved to Corynebacterium, then Actinomyces, and most recently Arcanobacterium. So yeah, if you've ever been reading an old case report and gotten totally confused, this is why. It wasn't until 2011 that it finally landed in its own genus, Trueperella. So what was the final straw in 2011? Why the big move? Well, it all came down to a perfect storm of genetic and phenotypic data. The 16S rRNA sequencing showed two totally separate family trees. And you know what backed that up? A super simple bench test, the CAMP reaction. The organisms that stayed in Arcanobacterium are CAMP positive, but our bug, Trueperella pyogenes, is CAMP negative. It's a really clean, practical way to tell them apart. Okay, so now that we know what it is, let's talk about what it does. Trueperella pyogenes kind of lives this double life. It's a notorious troublemaker in the veterinary world, but it's also an emerging and often really nasty threat in human medicine. Out on the farm, this thing is an absolute economic disaster. In cattle, it's famous for causing summer mastitis, which can just completely wreck a dairy cow's udder. It also causes uterine infections that tank fertility rates, not to mention liver abscesses. For farmers and vets, this bacterium is a huge, huge problem. And this is where it really hits home for us in the clinical lab. It can jump from animals to people, especially farmers, vets, or anyone who's immunocompromised. And when it does, the infections are no joke. We are talking severe, life-threatening stuff here. Infective endocarditis, brutal prosthetic joint infections, and deep, nasty abscesses. This is exactly why getting a good patient history is so critical. So you get a plate with this thing. How do you nail the identification and avoid the pitfalls? Let's walk through the key workup for it on the bench. Under the scope, it's a classic pleomorphic gram-positive rod. You'll see all sorts of shapes. Coccoid forms, club shapes, V forms. It kind of looks like a Corynebacterium. But, and this is a big one, there is a major pitfall. It decolorizes so easily. If you get a little too happy with your alcohol acetone, you can make it look gram-variable or even gram-negative, sending you down a completely wrong path. On the plate, give it about 48 hours in a CO2 incubator, and you'll see these tiny little pinpoint colonies. But the thing that really jumps out at you is that huge zone of beta-hemolysis. I mean, it's often 2 or 3 times bigger than the colony itself. That's its main virulence factor, pyolysine, just going to town. And of course, nothing's going to grow on the McConkey plate. Okay, the biochemicals are where we really start to separate it from the pack. It's catalase negative, right? So that's a great way to differentiate it from staph. It's also positive for gelatin hydrolysis, which totally makes sense given its ability to form all that pus. And here's a key clinical pearl. It's intrinsically resistant to sulfonamides. That's a super important piece of information we can give to the clinicians right off the bat. Now, these classic methods are great for a presumptive ID, but when the stakes are high and you need to be 100% certain, molecular is the gold standard. And for this bug, that means looking for one specific gene, the Pyogenes gene. So the Plo gene is the blueprint for pyolysin, that powerful toxin that literally punches holes in cell membranes. That's what's causing that crazy beta-hemolysis we see on the plate. And because basically every single strain of Trueperella pyogenes has this gene, running a PCR for it is an incredibly specific and sensitive way to lock down the ID for good. Okay, we've nailed the identification. We're feeling good. But now we arrive at the biggest challenge of all, the part that takes us from the bench straight to the bedside, antimicrobial susceptibility testing. And this is where it gets messy. So when we look at its resistance patterns, it's kind of a good news, bad news story. The good news is it's usually susceptible to beta-lactams like penicillin. Awesome. But the bad news? Resistance to pretty much everything else is super common. We're talking tetracyclines, macrolides like erythromycin, clindamycin. Resistance rates are often over 50%. This means you absolutely cannot guess. You have to do the testing. And here it is, the heart of the problem. This is a direct quote from the 2025 EUCAST guidelines. The bottom line is, there are no official EUCAST breakpoints. So what does that actually mean for you at the bench in a European lab? It means you can run the test, you can get an MIC value, but you cannot legally interpret it as susceptible at increased exposure, or resistant. This creates this massive divide in how labs around the world report this bug. In Europe, the EUCAST recommendation is to just report the raw MIC number and add a note telling the clinician to get an expert consult. Meanwhile, in North America, labs often use the CLSI breakpoints for coroniform bacteria as a kind of stand-in to give an interpretation. This inconsistency makes it almost impossible to compare data or track resistance trends globally. So what on earth do clinicians do? Well, it takes a really logical, step-by-step approach. For a really sick patient, they'll start with something broad spectrum. Then, once the lab calls with the ID and the MIC value, they can de-escalate to a targeted drug, usually high-dose penicillin or ceftriaxone if the MIC is nice and low. And you have to remember, treatment is a marathon, not a sprint. We're talking weeks, even months, for these deep-seated infections. This is all already super complicated in adults, but what happens when the patient is a child? With kids, it's even tougher. There is a huge lack of data on Trueperella Pyogenes infections in children. It's a massive knowledge gap. So treatment has to be very carefully extrapolated from adult data, with super careful dosing, and an infectious disease consult with a pediatric specialist is absolutely mandatory. This really underscores how much responsibility we have in the lab to provide the most accurate data we possibly can. All right, let's bring it all home. We'll wrap up with the most important takeaways for your daily practice. So if you remember anything from this, let it be these 4 things. 1, know the history. Its old names matter when you're looking at data. 2, trust that beautiful beta-hemolysis, but always confirm your ID, ideally with a Pyogenes PCR. 3, know the resistance patterns. It's born resistant to sulfonamides and loves to pick up resistance to tetracyclines and macrolides. And finally, number 4, report with caution. If you're a EUCAST lab, there are no breakpoints. Report the MIC and recommend a clinical consult. Just so you know, this entire explainer was put together using Gemini Deep Search, and the images you saw came from MicroCanvas. If you want to see a full list of all the source materials we used, you can check out the notebook document. We've put a link to it for you down in the description. And that's a wrap on our deep dive into Trueperella Pyogenes. But I want to turn the question over to you, especially those of you working in labs that follow EUCAST. How does this whole no breakpoints issue affect your daily workflow and your conversations with clinicians? I would love to hear about your experiences, so please share them in the comments below. And hey, if you found this useful, do us a favor and hit that subscribe button for more deep dives into the microbes that keep us on our toes. Thanks for watching.